So welcome back to the Classics of Immunology Journal Club. We've been into this for quite some time now, and we've gone through about 150 years of classic immunology papers. And so if you like this video, don't forget to like it on the website, on the YouTube website, and click on the little bell to get notified for more, because I do one about it, almost one a week. And then also check out my website and go onto the writing tab, because on the website, all of the classics of immunology are placed there. And so you can peruse the literature, Kendall. It's a good thing. So this week, we're doing a follow-on paper after last week when we did a Rosenthal and Shevach paper from the Journal of Experimental Medicine. It was published in 1973, and this paper was published back to back with that paper. But they're both a little bit involved, and I thought that rather than do them all at the same time last week, we should split them up. And of course, as I went into before, Ethan Shevach was just a young guy that had just finished his clinical training, and he found himself as a postdoc with Bill Paul at the NIH. So that meant he didn't have to go to Vietnam. And Alan Rosenthal was also but he was an independent investigator from a different department, different laboratory at the NIH. So, and Alan Rosenthal had been working on this idea that the macrophages contribute an awful lot to the lymphocytes in terms of their abilities to function. And while Ethan Shevach was coming at it at the role of macrophages in lymphocyte physiology and function from the standpoint of the major histocompatibility locus complex, genetic locus. At the time, if you go back into the literature, and the thing is that on the website now there's other papers, and I wanted to point you to the right ones if you want to read back into these, because I did not cover them. And these are all papers from the 60s. It starts with 1963. So that was 10 years prior to the paper that we're going to go into today. And it was from Baruch Benacerraf, who was at NYU at the time, and he was a young investigator in 1963. And he published, what he decided to do, you have to realize that they had just finally figured out the structure of antibody molecules in the late 50s and early 60s. And so the whole field of immunology had been focused so long on antibodies that Benacerraf decided that the next big question in immunology, was what constitutes an antigen? And so he started synthesizing long chain and branch chain polymers of amino acids to see what the minimum size would be, and to see whether or not, if he made an entirely synthetic antigen, it wasn't from nature, whether or not the immune response would recognize it and have an immune response. And so very fundamental question. And he found that the title of his first paper from 63 was Studies on Artificial Antigens, 1, Antigenicity of DNP polyL lysine and DNP copolymer of lysine and glutamic acid in guinea pigs. So that was 63, and that was really when he started exploring the nature of antigens. Then he had another paper out 3 years later, and these are all in the JEM. 3 years later, another paper from Benacerraf's group at NYU, The Behavior of hapten polyL lysine conjugates as complete antigens in genetic responder as haptens and non-responder guinea pigs. So he's into the guinea pigs. And basically there were only 2 strains of guinea pigs that were inbred. The rest of the guinea pigs out there in the big bad world were outbred, just like human beings in the big bad world. In an inbred strain is that so all of the animals from that strain are like identical twins. So, and in that paper in 1966, he defined for the first time that there was something funny going on with the genes, because he could study these 2 different inbred strains of guinea pigs that were identical, except for the fact that they were then defined by Benacerraf as being responders and non-responders or low responders in terms of their ability to mount an immune response to his synthetic antigens. And he called the genes responsible for this immune response genes, or IR genes. But the guinea pig was a problem because even though they're nice little docile animals, and even though they don't have a tail, so you can't pick them up easily, they only had these two strains, inbred strains, available. So it wasn't too long, but before in 1968, Hugh McDivitt from Stanford University in California decided to look at this whole question of immune response genetics in the mouse. And as you know, from 1930 on, George Snell at the Jackson Laboratories in Bar Harbor, Maine, had inbred and constructed all kinds of different kinds of inbred strains of mice. And the genetics had been worked out in the last 30, 40 years on all these inbred strains. And so he took advantage of the fact that he could do really much more sophisticated genetic experiments by crossing and so forth and so on, and then testing for antigenicity. And he had a paper out in 1969 68 in the Journal of Experimental Medicine called Genetic Control of the Antibody Response in Inbred Mice, Transfer of Response by Spleen Cells and Linkage to the Major Histocompatibility Complex. So this was very, very important. So because, as we've gone back with Peter Medawar in the 50s, it was clear that there was a locus in different mammals that conferred histocompatibility or incompatibility on the tissues of that particular strain. And to link the immune response to the MHC, major histocompatibility complex, was not necessarily an anticipated and straightforward thing. But the experiments that McDivitt put out in 68 and then another paper in Science in 1969 sort of nailed that down. So now that brings us up to the present paper from 1973, just 3 or 4 years later. And I want to preface what we're going to, before we get into this paper, it's one of the reasons to study the history of a particular subject, whether it's science or not science, is that you don't really have any perspective of how long things took to evolve so that the understanding changed or became a new understanding of anything. And so that's very, very important, because as I would always tell my students and postdocs, this is that anything's possible in science, but some things take a very long time. And so in this particular instance, we're talking about 1963, when Benacerraf first started studying these artificial antigens, until 1973, to the paper that we're going to look at today. The other thing that is the retrospectoscope that's interesting is so now it's 2021, and we know a lot more now than we did in 1973. That's sort of like 50 years ago. And what we know now, which is common knowledge in immunology, is that we know the molecular structure of the T cell antigen receptor. That wasn't worked out until the 80s, and so we'll get into that as time goes on. But now we know that the T cell antigen receptor is a multi-complex of several different genetic encoded proteins, two of which confer the antigen binding portion of the T cell antigen receptor, and then two others, which are called the CD4 or the CD8 molecules. They act as co-receptors, and what those co-receptors do, they're sitting right together with the antigen binding molecules, but they don't bind to to the antigen that's out there. They bind to the major histocompatibility complex encoded molecules, MHC encoded molecules. And that forms a very tight interaction on that whole thing. And then there's another set of molecules that are called the CD3 complex that contain the signaling aspects of the T cell antigen receptor. So that took a while to everybody to figure out, but now it's common knowledge. We all know that. The paper that we're going to look at today is some of the first data that began to show that this is what's going on. And it's really focused on the macrophage again. The title of the paper is The Function of Macrophages in Antigen Recognition by Guinea Pig T Lymphocytes. Number 2, number 1 we did before. Number 2, role of the macrophages in the regulation of the genetic control of immune responses. JEM 1973. So in the introduction, one of the things I wanted to say is that the authors of this paper are reversed from the previous paper. It was Rosenthal and Shevach. Now we've got Shevach and Rosenthal, and they both basically brought their about 50% to these two papers, and so it was a good separation or designation in the authorship. So in the intro, they make several points that they are going to focus on, and number 1 is they say that MHC linked to IR gene products, we're talking about the products of the gene, which would be proteins, are expressed on T cells in mice. And secondly, in guinea pigs, T cell functions, and those would be delay type hypersensitivity skin testing, and in vitro antigen induced T cell proliferation, and the carrier specificity of the B cell antibody production, immune response, are all recognized T cell functions. The third was that responder and non-responder B cells must be compatible with the T cells across the MHC in order to get antibody formation. And therefore, they come down and they say that the main functional role of the IR gene product is in antigen recognition by T cells. That proved to be true. That's very important. That's one of basically the bottom line of this paper. But then they go on to say in the introduction, they say, but the IR gene products might also be expressed by macrophages, which control and regulate T cell activation. Seems like a logical thing since that's what we know. And the last thing that they point out, and this is from the previous paper, allo-antisera against MHC gene products inhibit antigen activation of T cells. So they're sort of focusing down on this whole process and trying to figure out exactly what's going on. They end up saying this paper is going to deal with 2 questions. Number 1, can parental macrophages that lack the important IR gene products, can they activate parental T cells? And number 2, can allo-antisera block the macrophage T cell interaction non-specifically? I don't know what that means, but that's what they were wondering about. Or do they have to be blocking the IR gene products, or perhaps maybe both? This is what they focused down on. Now, the materials and methods in this paper are the same as in last time's paper, the previous paper we reviewed. We're dealing with guinea pigs, strain 2 and strain 13. Those are the inbred strains. And then they make the so-called F1 progeny of that by mating the 2s to the 13s. And so now you've got a combo of 2 times 13. Their antigens in this particular paper that they're studying, they have a copolymer of L-glutamic acid and L-lysine, and that has a molecular weight of about 115,000. Then they have DNP, GL, glutamic acid lysine. And then they have a copolymer of glutamic acid and L-tyrosine as another control of the whole thing. That has a molecular weight of 20,000. So I'm going to do some screen sharing here in terms of showing some of their data, and I'm not going to show all of the data. You're going to have to go and read this paper carefully, because I was afraid I'd get into the weeds and I would never get out again, essentially. I'm only going to show you 2 figures from this paper. So this is figure 1 from this paper. And as the figure caption says, this is stimulation of F1 lymph node lymphocytes for a source of T cells by parental or 2 times 13 macrophages that have been pulsed with DNP GL, DNP GT, or PPD. And the results are expressed as the differences between the control and antigen stimulated cultures, CPMs per culture, of tritiated thymidine incorporation. And each bar represents the arithmetic mean of 3 different experiments, plus or minus the standard error of the mean. And so we've got responding T cells that are 2 times 13. But when the antigen is GL, the antigen presenting cells, which would be the F1 macrophages, they lack when we're stimulating those cells with either F1 macrophages that have both 2 and 13, then we have a good response. And when we're using strain 2 macrophages, so that they only have strain 2, we get a diminution. And when we have 13 macrophages that lack the 2, and the immune response depends on having 2, is essentially very, very low. So then in figure 2 that I'm not going to show you, but I'll talk about it, the magnitude of the stimulation produced by the antigen pulsed macrophages from the non-responder parent is a lot less than from the responder parent. And they go on to do experiments to find out whether or not there's any antigen carryover, and they decide that's not the case. And they also do experiments to figure out whether or not antigen uptake by the macrophages that occurs in only 30 to 60 minutes, whether or not that's blocked by allo-antisera, and that's not the case. They go on then to look at allo-antisera, whether it inhibits antigen-induced lymphocyte proliferation, both by blocking the so-called macrophage lymphocyte interaction or by blocking the products of the IR gene. And they find it basically that it's both. Then they go on to what is shown in figure 5. So there's a lot of data. Here's the table and so forth and so on. So here's figure 5, and I only want you to look at this portion of the figure, because the other parts are just confirmatory of that. And basically what they've done is that they have, they're using T cells from the F1, 2 times 13, and they're using antigen pulsed macrophages from the F1, and they want to find out whether or not if they block either strain 2 MHC or strain 13 MHC, whether that makes a difference. And essentially down here, this is normal guinea pig serum here for this set of data, then strain 13 anti-2 essentially gets rid of everything, whereas strain 2 anti-13 only blocks and the 13 macrophages. And so essentially what's happening here is that they've concluded, and this is where they go into the discussion then, these data and all of the experiments that they did, the findings suggest that the macrophage lymphocyte interaction is mediated via the histocompatibility antigens itself, or something closely linked genetically to the MHC in the MHC locus that they couldn't separate genetically. And they go on to say then that the distribution of the antigen on the surface of the macrophage is probably random and unrelated to MHC encoded antigen. So they've got a thing about whether it's MHC itself or whether it's MHC plus something else. And they go on to conclude that during the lymphocyte macrophage interaction, the macrophages and the lymphocytes that share MHC encoded molecules come into very close functional contact. Now that's interesting. Exactly what that functional contact is the question. And they go on to say that the IR gene controlled antigen recognition sites on the T cell are physically related to the macrophage binding site, and both are linked to the MHC encoded molecules determined serologically, determined by their anti-sera. And that the IR gene product is closely related to MHC encoded molecules and or to the site of a macrophage lymphocyte interaction, which may be both the same, is what they're saying. And they go on to say then that this report does not establish whether or not the IR gene product is the primary antigen binding receptor on the T cell. So they don't know what the T cell antigen receptor is. But antigen recognition by T cells appears to involve a complex membrane unit consisting of an antigen binding site and a specific site for the macrophage lymphocyte interaction. So they're getting into this situation that are we dealing with one receptor or two receptors? And that's it. And they basically say that they need, that in the end, what we know at this stage of the game in 1973 is that you gotta have histocompatibility or T cell proliferation by antigen, for antibody formation, and that's another paper that's on the website from Benacerraf. And finally, another paper a year or two more recent, I think 74, 75, that said that cytotoxic T lymphocytes, their ability to kill the target cell requires histocompatibility. And that was by a couple of fellows by the name of Rolf Zinkernagel and Peter Doherty. And I should probably just mention offsides that Baruch Benacerraf was awarded a recipient of the Nobel Prize in 1980 for this work, for starting off this whole process. And he published a very interesting hypothesis paper in 1978 in the Journal of Immunology, where he proposed that what was really going on in terms of the macrophage component is that the macrophage was binding the antigen through the MHC. He said, I don't know what the T cell receptor is though. So then Zinkernagel and Doherty went on in 1996, 16 years later, and they got the Nobel Prize for their contributions. And you should go on read all of these papers and go on and make up your own mind as to exactly who deserves credit for what. So if you like the Journal Club and if you like this video, or if you don't like it, you can send me an email. But if you like it online on the YouTube website and click on the bell and visit all of the other, so my own website and the social media sites that deal with immunology, and particularly the classics. So it's been great. Thank you.